Welcome to the deep dive. We're here to sift through a lot of research on one really complex topic and, well, make sense of it for you. Today, it's Autism Spectrum Disorder etiology, understanding the causes. And if you've looked into this at all, you know it's not straightforward. It's definitely a puzzle.
It really is a puzzle.
Huh.
And our job today, our mission, is to try and bring some clarity. We've pulled together findings from major reviews, some very new studies too. We're looking at the genetics, the environmental factors, and critically, how they interact. That's the gene by environment piece, the GXE. The idea is to find where these different lines of evidence converge, maybe hinting at ways forward for therapies or even prevention.
Okay, so let's unpack that. The sources point clearly to two main areas of risk, right? There's the genetic side, strong, definitely, but complicated. And then there's this growing body of evidence about environmental factors, especially things happening before birth, during development.
Exactly. In utero. That seems to be a really critical period.
All right, let's start with the genetics foundation. We know ASD runs in families. The risk for a younger sibling, it's significantly higher than the general population, around 2 to 8%, I think.
That's right. And it jumps even higher, maybe up to 12 or even 20% if you include siblings with broader, maybe milder traits.
And twin studies have always shown a strong genetic hand, right? Heritability estimates used to be thrown around like 80% sometimes.
Yeah, historically very high estimates. Though one big recent study put it closer to 55%, still substantial, definitely. But here's the thing, even with that high heritability, finding the specific genes involved, that's been incredibly challenging.
Why so tough?
Well, partly because ASD is so varied clinically. The spectrum is wide. Two people with the diagnosis can present very differently, and their underlying genetics can also be quite different. Plus, the inheritance usually isn't simple Mendelian genetics.
Not like brown eyes or blue eyes, passed down simply.
Exactly. It's complex, involving many genes. Researchers initially looked hard at common genetic variants, but those usually only have a tiny effect individually, like odds ratios barely above 1. So the focus shifted, quite smartly, towards rare mutations, and especially de novo mutations.
De novo meaning new in the child, not inherited.
Precisely. Brand new genetic changes. And that shift has been much more fruitful.
Okay, so what pathways are these rare and de novo mutations pointing towards? Is it all over the place, or is there convergence?
There's definite convergence. Thousands of different genetic alterations have been found, but they seem to funnel into a limited set of biological jobs. Synaptic function comes up again and again in the forces. Think of synapses as the connection points between brain cells.
The communication hubs.
Right. So genes involved in building and managing those connections, like NLGN3, SHANK3, NLGN4X, are frequently implicated. We also see pathways involved in how cells grow, certain signaling pathways like GT-pathways, and neurogenesis, the creation of new neurons.
And it's not just which genes, but when they're active, that seems crucial.
Absolutely critical. Both the common variations and these rarer mutations seem to exert their main influence during a specific developmental window.
Corticogenesis.
That's the formation of the cortex, the brain's outer layer.
Exactly. During fetal development, it suggests that if the genetic blueprint is slightly off during this critical construction phase, the brain's wiring can be affected right from the start.
And connecting this back to things we might be able to influence, like diet or supplements, the sources flagged a few specific gene variations, didn't they? Polymorphisms.
Yes, some umbrella reviews found suggestive evidence for variations in genes like MTHFR, that one's involved in folate metabolism. Also CNTNAP2 and VDR, the vitamin D receptor gene. It's suggestive, not definitive, but it connects genetics to metabolic processes potentially influenced by environment or nutrition.
Interesting. Okay, so the genetic stage is set early during brain formation. That leads us directly to the environment during that critical window. What's happening around the developing fetus? Let's talk about the maternal environment and factors around birth.
Yeah, it seems clear that the processes underlying ASD often start in utero. One well-established factor is parental age. Advanced maternal age, say 35 or older compared to mid to late 20s, comes with about a 1.4 times higher likelihood, roughly a 40% increase. Advanced paternal age is a factor too.
And beyond just age, the mother's health during pregnancy seems really important, especially metabolic and immune health.
That's a major theme. Conditions like gestational diabetes show up consistently. And the potential mechanism there is fascinating. One study found mothers with type 2 or gestational diabetes were almost 3 times more likely to have anti-fetal brain autoantibodies.
So their immune system might be creating antibodies that could affect the baby's developing brain.
That's the hypothesis, yes. It suggests an immune component crossing the placenta, and this ties into the broader theme of maternal immune activation. Things like maternal infections or even just running a significant fever during pregnancy. Those are strongly linked to increased risk in the child.
The fever itself, or the infection causing it.
That's the crucial question. And one study had a really striking finding. When mothers took anti-fever medication, it seemed to reduce the subsequent risk of ASD in the child.
Wow. So that points more towards the inflammatory response, the fever reaction, rather than the specific bug.
It strongly suggests, yes, that the inflammatory cascade might be the key mechanism. It's a huge area of research now. We also see links with maternal autoimmune conditions like type 1 diabetes or rheumatoid arthritis. Those showed about a 60% increased odds ratio.
Okay, so immune activation, metabolic issues, age. Are there other factors mentioned?
Yes. Things like birth order, being firstborn versus third or later, showed a slight association. And prenatal use of certain medications, specifically psychoactive drugs, were linked to about a 60% increased risk as well.
Important to mention, too, what didn't seem strongly linked. Sometimes that's just as revealing.
Good point. The sources specifically found pretty strong evidence against an association for things like treaties fetal loss, preeclampsia, maternal hypertension, and swelling during pregnancy. Helps narrow things down.
Right, focusing the search. Okay, let's shift from the internal maternal environment to external factors, pollutants, toxins, things we might breathe or ingest. The big concern here is that the developing brain is so vulnerable, and these things can get past protective barriers like the placenta or the blood-brain barrier.
Exactly. The developing nervous system just doesn't have the same defenses as an adult's. Even low levels of exposure can be problematic. And meta-analyses are consistently finding links. Air pollution is a big one.
Like traffic fumes.
Yeah, things like nitrogen dioxide, NO2, carbon monoxide, CO, those show significant associations. Particulate matter too, especially the really fine particles, PM2.5. Exposure during pregnancy seems particularly risky.
What about other types of environmental chemicals? Metals, maybe? Plastics?
Heavy metals in general show an association. Elevated levels of cadmium and mercury are often reported in studies involving children with ASD. And then there are the endocrine-disrupting chemicals, EDCs. These mess with our hormone systems. Phthalates, which are everywhere in plastics, cosmetics, show up in these studies.
There was a real-world example in the sources, wasn't there? An ecological study in Italy.
Ah, yes, the Taranto study. Very striking. Taranto has a huge industrial steel plant. Researchers basically compared kids living in the high-pollution areas near the plant versus those in cleaner areas of the same region. And they found a significantly higher prevalence of ASD in the kids living in the high-risk industrial zone. It really drives home the potential impact of localized heavy pollution.
So how do these pollutants actually harm the developing brain? What's happening at the cellular level?
A key mechanism seems to be oxidative stress. You can think of it like biological rusting. These toxins can damage cell membranes, interfere with enzymes, and trigger the production of damaging molecules called reactive oxygen species. This process hammers mitochondria, the cell's power plants, and fuels inflammation. So it connects right back to those immune and metabolic stress pathways we were just talking about.
It all seems interconnected.
Very much so.
Which brings us neatly to the gene-X-environment interaction, the GXE effect. Because it's probably not just the gene or just the exposure, it's how your specific genetic makeup handles that exposure.
That's the crux of it, yes. Differential susceptibility. It's an idea that gained traction when researchers noticed something interesting about siblings. Unaffected siblings of children with ASD tended to have experienced fewer complications around birth than their affected sibling, but still slightly more complications than kids from families with no history of ASD.
Implying the unaffected sibling might have had a similar exposure but handled it better genetically.
Exactly, suggesting the child who developed ASD had a genetic vulnerability that made them less resilient to that particular perinatal stressor.
Can you give a more specific example from the sources, like a gene interacting with a toxin?
Sure. Animal models show this quite clearly. If you have mice with genetic mutations affecting synaptic function like those neurologen mutants we mentioned, they become much more sensitive to things like oxidative stress or mercury exposure. There's also evidence suggesting specific pollutants might interact directly with the genome. For instance, one type of PCB, PCB95, might influence the number of copy number variations, deletions, or duplications of DNA, specifically around a region on chromosome 15 that's strongly linked to ASD.
Wow, so the chemical could actually be tweaking the genes, or at least their effects. And speaking of PCBs, those persistent industrial chemicals, did the sources flag any specific ones?
Yes, one particular PCB, number 138, showed up with a pretty strong association. Risk was almost doubled, an odds ratio of 1.84.
Okay, now for something really new from the sources, microplastics. This feels very current.
It is very cutting-edge research. There was a mouse study discussed where they exposed mice to polyethylene microplastics. It's a super common plastic both before birth and afterwards. And the exposed mice developed behaviors resembling ASD symptoms in humans. Things like reduced social interest, more repetitive actions.
And how? What was the mechanism they found? It wasn't just the plastic itself lodging somewhere, was it?
No, it seemed to be mediated through the gut. This is really tying things together. The microplastic exposure dramatically changed the mice's gut microbiome, their gut bacteria.
Ah, the gut-brain axis again.
Precisely. They saw a decrease in beneficial bacteria like Lactobacillus reuteri, which other studies have shown can actually reduce social deficits in mice. And at the same time, they saw an increase in other bacteria like Alistibes putridinus, which have previously been found to be more abundant in some children with ASD.
So the pathway is environmental toxin, microplastic, disrupts the gut bacteria, influences brain development and behavior.
That's the chain of events suggested by this study, yes. Environment, gut, brain, all linked. Hashtag tech synthesis and conclusion.
Okay. So if we step back and look at the whole picture, the genetic risks focused on synapse development during corticogenesis, the maternal immune activation, the air pollution, heavy metals causing oxidative stress, even microplastics messing with the gut microbiome. What's the big takeaway message for you?
I think the overwhelming message is that ASD is truly multifactorial. It starts with a genetic vulnerability, often affecting core pathways like how neurons connect and develop or how the body handles inflammation. But then environmental factors act upon that vulnerability. And the timing seems absolutely critical. Those prenatal and very early postnatal periods are windows of heightened sensitivity. The same exposure later in life might have a very different or much smaller impact.
Given how complex this is and how much detail is emerging, where does the research need to go next to really crack this?
Well, we definitely need more prospective studies. Following people over time, measuring exposures accurately as they happen, ideally using biomarkers, not just relying on memory years later. And crucially, these studies need to be genetically informed. We need to look at specific exposures in combination with specific genetic profiles to really understand that GXE interplay. That interaction is where the leverage might be for prevention or early intervention.
That makes a lot of sense. This has been an incredibly insightful deep dive. Thank you for walking us through such complex material.
My pleasure. It's fascinating, albeit complex territory.
Absolutely. And now, as always, something for you, our listener, to think about. Given this emerging picture where specific pollutants like microplastics or maybe heavy metals seem to exert their main effects by interacting with known genetic weak spots, how close are we potentially to a future where managing ASD risk isn't just about general public health advice, but involves more personalized strategies? Could we see things like genetic screening paired with targeted monitoring for specific environmental exposures relevant to your genetic profile? Something to consider. We'll see you next time on The Deep Dive.